I'm very pleased to introduce our next speaker, Dr. Andy Cadi, who's from the Institute for Regenerative Biology and Medicine in Munich. And Andy is part of a group that, there are very few groups worldwide who are working on developing cell lines for marine invertebrates. And Andy's group is one of the few. Our group focused on sponges at one end of that phylogenetic spectrum. Andy's group has been focusing on tunicates at the other end of that invertebrate phylogenetic spectrum. And he's going to talk to us today, a very apropos topic, especially since this afternoon we're having a panel on, how do you get the best medium, optimization of medium. And so Andy's going to talk to us today about some groundbreaking work they've done on developing novel media for primary cell cultures of the tunicate Botrytis schlosseri. Andy?
Thank you. I thank the organizers for inviting me here to speak about, it's this or this? To speak about improved media formulations of primary cell cultures of Botrytis schlosseri. I need to synchronize this both. Right? Now only this. Yeah. Huh.
You might want to put your lapel mic on the other side, Andy.
Sorry?
Put your lapel mic on the other side.
So as you all know, the capability of cells to live under in vitro conditions. You cannot hear me? The capability of cells to live under in vitro conditions is an important tool in a variety of scientific disciplines. Marine invertebrates may be regarded as a major source for such application, comprising more than 30% of animal species and covering more than 20 different phyla, which representing a rich source of cell types. And many marine invertebrate cell types exhibit a broad range of cellular potentialities such as multipotency and totipotency. Until recently, there is yet no single permanent cell line available from any marine invertebrate because of the cells are stop dividing in vitro 72 hours within after their isolation. However, a recent development in the sponge cell cultures have highlighted the importance of established nutrient media that eventually was the major of publications, showed the successfully long-lasting cultures of several species of sponges. Botrytis schlosseri is one of these marine invertebrates that several studies attempted to develop primary cell lines and primary cultures from Botrytis, but these studies describe the general in vitro conditions in these cultures, and they failed to show all the requirements that we need for these cultures. Botrytis schlosseri is an important model organism in a variety of scientific disciplines, including stem cell biology, because many cell types from Botrytis exhibit a broad range of cellular potentialities, including multipotency and totipotency. It belongs to the phylum Chordata, which is the closest living invertebrate that is belong to the vertebrates. It has two reproduction modes, the sexual and the asexual. The sexual reproduction mode starts with egg fertilization and goes through the classic embryonic stages, featuring chordate characteristics such as tail, the notochord, the muscles, the neural tube. And then the larvae settle down near the mother colony. The tail undergo apoptosis and creating the oozoid, which is the first module of the animal. Colonies that are developed from the oozoid form a star-shaped groups of zooid called the systems. The systems are embedded to each other through a common blood vessels, which have ampullae at the end of the colony and in between the zooids. And each zooid possesses an oral siphon and an atrial siphon for each system. The asexual reproduction mode is a development cycle that happens every week where the animal grows through budding. Okay? Oh, sorry. Through budding, where it's culminated at the end of this stage by apoptosis, where each zooid absorbed and the primary bud taking the next stage of the developing zooids. The Botrytis cells have been grouped into their differentiation pathways, and the undifferentiated cells, which are the hemoblasts, were the source of these three lines. The first two involved in the defense response are represented by the macrophages, the granular amoebocytes, and the morula cells. And the storage cells are involved in the catabolite and excretion processes in the animal. Up to date, there is no single cell line available from Botrytis. And it is repeatedly shown that the extracted cells are stopped dividing in vitro within 72 hours from isolation. And also many of the cultures are associated with the Strauss-Zochetrids. The Strauss-Zochetrids are a group of unicellular protists belonging to the kingdom Chromista, and they dominated the cell cultures, and then by day 8, only axenic cultures of these organisms can be developed. And according to this, we aim first to characterize the Botrytis cell types in vitro, to understand the condition required for them to grow by utilizing 5 media formulations, and to provide an assessment about the dominant cell type, the viability, and the proliferation of these studied cells in the media. These aseptic methods have been used in all the cell culture experiments, where the cells are extracted from the marginal ampullae of the animal, and this was proven to reduce 90% of contamination, including these Strauss-Zochetrids that are developed. Oh, I need to synchronize this. So, first, we wanted to see which types of cells are developed in these cultures. And the identification of the cells was done by histological, nuclear, and membrane staining. And we characterized these 6 types of cells within 3 lines. Also, we characterize the position of the nucleus and the membrane of the whole cell. Because there is lack of information in the literature about these developing cells in the Botrytis cultures, and also it contributes to our understanding how they are growing, which cells are dying, which cells are proliferating, and also it also confirmed it also confirmed previous studies on in vivo studies. Oh. So we started to see how they are growing. For this, we used 40 animals from a variety of blastogenic stages with high viability at the beginning of the studies, and we cultured them with media 1, 2, 3, 4, and 5, TGM1, TGM2, and TGM3, 4, and 5. TGM is tunicate growth media. Starting with TGM1 media that was formulated containing basal media without artificial seawater. We see that the cells start to lose viability 6 days after cell culture initiation, with hemoblasts, pigment cells, macrophages, and morula cells being the most dominant cell types in the cultures, and PCNA positive cells were recorded for hemoblasts and macrophages. TGM2 media was formulated also containing basal media but without artificial seawater, and also cell viability started to decrease 6 days after cell culture initiation, with hemoblasts, macrophages, morula, and pigment cells being the most dominant cell types in the culture, and positive cells for PCNA were recorded for the hemoblasts, pigment cells, and the morula cells. TGM3 media, on the other hand, was formulated containing only artificial seawater without basal media, and viability started to decline 4 days post cell culture initiation, with morula cells and pigment cells being the most abundant cell type in the cultures within the 8 days of from initiation, and positive cells for proliferation were recorded for hemoblasts and macrophages. When we see all this, we see that there are no scientific evidence culturing Botrytis cells without artificial seawater or only with basal media, and despite this, various levels of cell types proliferated. Therefore, it is reasonable to assume that the media TGM1, TGM2, and TGM3 are still inappropriate or lacking of essential factors like media and basal media or artificial seawater. And despite this, cell proliferation was achieved, even that we know that Botrytis cells stop dividing in vitro within 72 hours post cell culture initiation. TGM4 media was formulated containing different proportions of basal media and artificial seawater, and cell viability started to decrease 18 days post cell culture initiation, with dominant cell types composed of pigment cells, hemoblasts, and macrophages without morula cells at the first week of the culture, and positive cells for proliferation were recorded for the hemoblast and the morula cells. And TGM5 media was formulated containing the same proportions of basal media and artificial seawater, with viability that started to decrease 16 days post-cell culture initiation, with hemoblast, macrophages, and morula cells being the most dominant cell type in the culture, and pCNA positive cells were recorded for the hemoblast and the macrophages. As an overview for these, we saw, for these two media, we saw that there are longer cultivation periods that observed for TGM4 and TGM5. And this is also confirming previous results on short viability study on coral cells that showed a decrease in viability of from 17 to 30 within the first week. And here we cultured them for 4 weeks, almost 1 month. And various levels of cell proliferation were observed for the hemoblast, for the macrophages, for the morula cells, and for the pigment cells. To conclude that there are no scientific evidence revealing the proliferation, the durability, and the cell type composition of Botrytis cells under prolonged in vitro conditions. Thus, the findings illustrate that Botrytis cells can grow when the media is altered, exactly as it has been shown in the sponges cell culture, where they changed the media, they showed proliferation and high viability of the cells. Oh. And also the results are further demonstrating that it is possible to grow the Botrytis cell types in a consistent and reliable manner. And the novel findings and the knowledge that we obtained from this study, improving the in vitro conditions for the Botrytis cells to grow, and may lead to the creation of cell types with specific media. And basically, I didn't put it here, but this is what I'm doing now, and I already spoke about this. I have some 3 cell types from Botrytis that are growing independently and not with the whole population of the cells. And with different media formulations, because these media were formulated only with the proportions. We played with the proportions of the components, but this new media, we add also growth factors and many other goodies that the cells need to grow, and yeah, soon to be published. So, and that's it. Thank you.
I was wondering if you have done any maybe molecular analysis of different population of cells.
Sorry?
Have you done any molecular analysis of 3 different population of cells besides histological staining and observation?
Yes. I mean, molecular analysis, you mean the identification of cells?
Any biomarkers in terms of one is macrophage-like and so on?
So
Maybe RNA-seq, maybe?
Yeah, the thing is that not like all you guys, for us, we don't have markers. And we are basically in this tunicate and also in marine invertebrate field, we are embracing what we already know from mammalian cell cultures, and you know, and then we are going back to their genome to check all these markers. We did some antibodies from these species, but yeah, we are going to this point. But we tried. Now we need to publish some articles about metabolomics, single cell. Yeah. Uh-huh.
To always refer to mammalian markers.
Yes.
So it's good.
Hi there, thanks for your talk. I'm wondering, because you're diluting your media, do you have to change your carbon dioxide concentrations in your incubator? And then secondarily, have you tested any different oxygen levels for your cells to see if they prefer more or less?
Very important question, but they are growing in atmospheric conditions.
Oh, but you're using like DMEM and stuff, but no.
No.
Oh, interesting.
But if you saw, the media formulations contained a lot of HEPAs.
Oh, so that buffers.
So to compensate that, I put more HEPAs because I'm not adding also oxygen.
Interesting.
Yeah.
All right, thank you so much.
We have time for another question.
Thanks, Andy. That was very interesting. We're pretty excited to see what the next phase is going to be. So one of the breakthroughs for us was when we moved away from our lab rats, our regular model species, and you've been using Bacillus schlosseri for a long time. Have you tried any of these on other species of tunicates?
Yes, actually, I'm in my phase when I'm trying all these on other species. So I'm trying on Botryloides. This is the cousin of Botryllus. I'm trying on coral cells. And I'm also trying to fuse between fish cell line and also Botryllus cell line. And to make it, well, I don't want to come with bombastic things like immortalize. Yeah.
We have time for other questions. Please.
It could be, you seem to allude to the fact that you do have some cells growing individually, and so I guess you're about to publish this, so you probably don't want to give too much information, but are you thinking that these are growing in a continuous fashion, or you can't talk about that?
No, I can talk, of course. One of the things is that in these articles, which I presented today, we don't know actually if these are the contribution of the proliferation is from the dead cells or because we have a population of many cell types. So we don't actually know if they are from the dead cells and some of them proliferate. So we can say, okay, the morula cells and the pigment cells and the macrophages are proliferating at this stage. Now we know that because when we grow them separately, we know that some of them have doubling time of 32 hours, some of them have doubling time of 48 hours, depends which cell type. The one that I'm trying to grow now, 7 months already, they are growing in cultures separately from the whole population. I started from one single cell, like limiting dilution, and then growing to the phase, because I want to make sure that they are not. No, it's not going back. They are not these Strauss-Tochitrids that are growing there in the culture. So, yes, for your question, yes, they are growing. their capacity, and some of them, they are also reaching a plateau and then starting to die.
But nothing is quite immortal yet?
Usually to make cell immortal, you need to induce things.
So it's not spontaneous in marine invertebrates?
It depends. It depends. It can be spontaneous. I don't know how to answer this. Not because I don't know. The cells are acting different in different conditions. So I don't know.
Thank
I would
you,
like
Andy.
to know the answer, of course.
Excellent. Thanks so much, Andy.
Welcome.
Well, it sounds like we've got a couple more minutes, so if I may I ask a question? Or are there any other questions? Yes, okay. So I'm not sure if you caught, but as Cindy introduced, so I'm on the fish side in terms of culture, but also more on the toxicology side in terms of exposures, as sort of Anita alluded to. So I guess my question is, has anyone, either in your lab or other labs in terms of these tunic cultures, done...
From tunicates.
Yeah, from tunicates,
Yeah.
have kind of delved into what happens when these cells are exposed to potentially harmful substances, those toxicants. Because looking at like marine environments and compounds like oxybenzone that comes off of sunscreen, impacting tunicates in those those areas. So has anyone kind of done that? I've seen it for coral,
Yes.
but I just didn't know if anyone's done more
Yes,
cell.
yes,
Yeah.
yes.
Okay.
Thank you. This is an important question, also for the field. The thing is that Botrytis, what I showed here, this is something else. Botrytis schlosseri is also invasive species. So, as I said, the cosmopolitan Botrytis schlosseri, I think this is very important for the field, because if this, and let's talk for real, if this animal is so successful, it means that its cells has a lot of variety, okay, of exposures, let's say chemicals or contaminant or whatever, and not so much done on this. I know that they did radiation. This is one of the examples, different kind of radiations, UVB, UVA, even that it doesn't, UVC, sorry, even it doesn't penetrate to the atmosphere from the, and also some antioxidants, and that's it. But this, I think it's very important. But of course, after we have, hopefully, a cell line, we can try all this, and then I can tell you what happens, especially with the pollution at the sea, which is important.
Wonderful. Yeah, I think we do have time for one more, we do have time. So for questions.
I have a very unhealthy obsession with cell morphology, so please excuse me. The macrophages, what morphology is it? Have a specific share of cell morphology or shape? Does it have any like veil-like projections around the edges? Or?
Yes, so they, of Botrytis you mean, not the fibroblasts of human, mouse and all this. So the Botrytis, they have, first of all, they are circulating in the bloodstream. So they are, they can go everywhere to the Botrytis tissues. They are big cells varying between 15 to 20 micrometers diameter, like the whole cell. They contain vacuoles, and they are involved in the defense response. So you have, especially with Botrytis, you have, when two colonies meet each other in the field or in the laboratory conditions, they have two options: to fuse, and cells are migrating from one colony to another, and to reject themselves. And this is connected to their DNA, and I'm not going through it. So the first defense line when they reject themselves are the morula cells, which I showed here, and the macrophages. So they are big cells that are basically protecting the Botrytis from any also pollutions from outside.
It's wonderful. Thank you. Wonderful.
Yeah, Surjeet, he's right over there. Yeah. Begin again at 12:30. So for Surjeet, you can start walking up. Anyways, Dr. Surjeet Arya, he's from the University of Kentucky. He's been working in Reddy Polly's lab. He's been there for a couple years. And I had the honor of working with him when he first came to the US from India, but it was during COVID. And I was in a USDA lab, and we could not authorize him to come and actually work in our lab. So we worked together virtually for at least a year and have continued to do so. But I've been very impressed by all Surjeet has been able to accomplish. And so I'm very excited to welcome him here to the podium and to give his talk on what he's been doing really with midgut, insect midgut cells. So here we go, Surjeet.
Thank you so much. Okay. Hello. Hi. What's the time right now? Yes. Hi. Good morning, all of you. Myself, thank you so much, Dr. Cindy Winman, ma'am, for introducing me and giving me the opportunity to present here. And currently, I'm going to present this work which I did last year. And this is a part of not actual work what I was doing, but it is kind of work I'm going to use it for my the future work. The work I have finished it. So this topic I'm discussing here today is unraveling cellular heterogeneity in insect midgut cells and their implication for agriculture and beyond. And this is our research team, myself, Sujit Kumar Arya, working as a postdoctoral research associate under supervision Dr. Reddy Palli, in collaboration with Dr. Doug Harrison. And before going into detail about my project, my work, I will give, this is the introduction I'm giving on this topic. Everyone knows this cellular heterogeneity refers to the variation in cell characteristics within a seemingly uniform population of cells, and this variation can be in terms of gene expressions, protein levels, metabolic activity, and cellular behaviors. And because I'm working on midgut, so my target was to look out for the midgut tissues. So as we know, in tissue like in the midgut, heterogeneity is crucial for the functions. We know this from the literatures because they have different cells to undertake a specialized roles. And what is the significance of this midgut is that this midgut has provide a key site of nutrient absorptions and metabolism and exhibiting a complex landscape of the cellular types and different midgut cell types, and understanding this diversity is essential for insight into normal gut function and potential insecticidal targets, because we are currently working in collaboration with the CAMTAC. So they were interested in looking for the insecticidal targets. And as we all know, this is the conference for In Vitro Biology, so it is mostly related to cell lines. So I presented here my cell lines work. So as we all know, insect cell lines have become indispensable tools in molecular biology, virology, and bioprocessing research. And however, understanding and managing cell heterogeneity within these cell lines is crucial for maximizing their utility. Like most of the people have talked about their using in for meat productions and for other testing the insecticidal target and toxicity assays. Recently, not recently, but in 2027, Hidalgo et al. Used live cell imaging techniques to facilitate the identification of district phenotype within a population of baculovirus infected SF9 cells, because we know that these cells are, we all were saying this is immortalized cells, and they were fast growing cells. When I started working on 617 cell lines, I found them most fast growing cells among all other cell lines. And I was very interested to know that why they are growing very fast, and what is the reason behind they are growing so very fast. At that moment in time, I was learning, because I learned a lot of things from my ma'am, and she taught me a lot of. I have worked on 8 different insect cell lines. So this is one of the cell lines I worked on. This is 617 cell lines. So we don't know about whether this cell culture is homogeneous and heterogeneous. So to reveal that things, we just go ahead with this 6-7 cell lines as a proof of concept. And many people, like as I saw Hidalgo et al. In 2017, used live cell imaging techniques to understand the heterogeneity in this baculovirus infected cell, SF9 cell. And other research was done on Zhao et al. In 2021. They used CRISPR-Cas9 screening as well as single-cell RNA sequence analysis to unveil genetic heterogeneity in this, what is called pancreatic cell line, which is MIN67 cell lines. And these findings challenge the assumptions of homogeneity within cell lines, and individual cells within the populations can exhibit characteristics, behaviors, like I'm going to show you later. So we have this popular technologies we were using long back was RNA-seq analysis for analyzing for differential gene expression analysis. But the reason we were using before was like, we don't know we are just accumulating this everything in just pooling them, and we are just getting the differential expression from whole the cells. We don't know which cells is expressing which genes and how they are differentially expressed between the cell types. So the drawbacks of using the bulk analysis is just that they are masking the actual heterogeneous differential expression, which is mostly expressed by the different cell types. So this single cell technology helps us to revealing out this heterogeneous nature of the homogeneous the so-called homogeneous population in heterogeneous populations. And in this single cell analysis, we have some barcodes. And we are using these barcodes to label, we providing them as a separate UMI label identifier, and we separate the cells into different different like clusters. And this cell cluster can be later on, like after analysis, we could able to see them as a different cell types and with a differential expression from individual cells. And we could able to, whatever is masking during the bulk attack can be revealed using this single cell analysis, which so that we can subpopulation expressing variability of thousands of cells can be revealed out, and we can understand how exactly something is happening in the cell type as well. So, okay. So my objective of that work was, first objective was to optimize method for single cell sequencing in mid-grid cell lines, which is SFMG0617. I get the cell lines from USDA labs. And why I started this single cell work with this cell line is because the current project was on for mid-grid tissues. And we need to optimize this single cell pipelines, because currently I'm a bioinformatics person and I'm a little bit of molecular biology. So, my first task was to optimize the single cell complete pipeline from the like tissue isolation to the analysis. So I started with this mid-grid cells first, and then second objective was to perform single cell sequencing using NovaSeq platforms. And the objective third was to isolation and enrichment of distinct cell types in the mid-grid cell line, and the testing the reproducibility and standardizing the analysis pipeline. So this is my single cell RNA-seq pipeline that I have standardized for mid-grid cell lines. First, we grew the cell lines, like I said, is in insect culture medium. When the cells were like 60 to 70 or 70 to 80%, we collected the cells and passed them through the filters. And with, this is the protocol we followed, like cell isolations and debris removal, and wash and resuspension, cell resuspension, visualization, and counting. When we see cell viability more than 90%, and we count the cell, because this single cell platform what we used was 10x, and they have a limitation of we can capture only 10,000 cells. We capture 10,000 cells with this. And after capturing the 10,000 cells, we sequence them using Nova 60,000 platforms. And after this platforms, this sequence, we got the sequence reads, and we processed using the cell ranger pipeline and filter these cells using the quality matrix. Like we have some parameters like to filter out the low or bad quality cells. And then after that, this is cell ranger is for the raw output, raw file processing things. And then we use Surat package, which is a R-enabled package for analyzing this single cell data set. And we further cluster these filter counts using Surat. And then we use find marker function to identify the marker genes to label the individual cell types. And then we perform the trajectory analysis, which is using the Monocle 3, which is a kind of computational framework having additive models that help us to see how the cells are differentiating during the pseudotemporal progressions. So this is output results of the single cell sequencing data from the cell rangers. So the first parameter is sequencing. We generated 2 replicates of the single cell data sets, the replicate 1, replicate 2. And we got mean reads more than like 30,000, which is said to be a good for single cell data set. And then the valid UMI's valid barcode was more than 97%, which said to be a good for this a good data set. And then mapping the second sequencing parameter, which we saw that like read map to the genome was more than 92%. And the third parameter was the nuclei. The estimated number of cells was like 10,000 for the first replicate, and more than 10,000, and 8,460 for the second replicates. And median genes per cell was found to be more than 2,000 and 2,513, which is said to be an optimum number of median genes we need to have for a good single cell data set to be further taken for the downstream analysis. The total genes detected was like 14,892 for the first replicate and 14,984 for the second replicate. And this is how we visualize the single cell data set using a TSNE plot. We have another another algorithm which is UMAP, but here I'm presenting because cell ranges generated both this TSNE plot basically. And this first replicate, as you can see, this is a raw clusters. As what we are assuming before was we were assuming that is a homogeneous population, but now we can see we are seeing a different cell clusters. And so it's not a homogeneous population, it could be a heterogeneous. So we are going to validate it later on with the marker genes. So here you can see a heterogeneous population of the cells, and we could able to identify like 14 clusters, which is a raw clusters in the replicate 1, and 13 clusters in replicate 2. And this is the percentage is the number of cells we identify in each cell cluster set. And this is our standardized pipeline for analyzing single cell data set, starting from the filter for generating this. This is called feature RNA and feature count RNA and percentage empty volume plot, which it shows you the quality of the cells. When you there is some parameter, there is the numbers written on the y-axis for all this and feature RNA, and count RNA, and percentage empty. So we have a threshold parameters, like we should not go, it depends on the data set. If your cells have more mitochondrial genes, you should have like 10% sweat parameters for this one, 10% of the mitochondrial rates, and some of the data I have seen 30, they are using 30%. So it all depends on the data set you are working on or the tissue you are working on. So we have different parameters for every data set. So we set some parameters over here to filter the low or the quality cell or the cells which fall below threshold values. After that, we got the cells, and this is the process. Once we get the data, this data I have analyzed using Surat, which is a single cell pipeline to analyzing a single cell data set. And after that, we normalize it. This is the process. We go first we normalize the reads, then scale it, and then do dimension reduction using principal component analysis. When we analyzing the data, we could able to see a lot of principal component. We need to normalize. We need to identify some minimum principal component that is giving a good like distribution of the cell clusters. So after that, we visualize the fourth step is the visualization using UMAP. As we can see, first initially I showed you like in Cell Ranger we were seeing 13 and 14 cluster replicate 1, replicate 2. What we did, we merged them together, both the data set, and as you can see here, we would able to see here 10 clusters, and this 10 cluster is a good cell representing clusters, and then we remove some doublet. Doublet is actually when 2 or more cells representing not representing a single cell information, but they are representing 2 or 3 more cells representing a single cell information. We need to remove them. We consider them as a low quality cells or the cells which is not supposed to be taken for the downstream thing. So we removed it. There are multiple programs available currently to remove this. I use doublet. And that helped me to, I set the parameter, the number, the 10% of the 7.5% I used for my 617 cell data set to remove the doublet. And after that, we removed doublet. The green, as you can see, the green color, sorry. Oh, sorry. The green color one is the singlet, and the little bit dot of here is the doublet, is a red color. Yeah, this is the red color, the doublet. And once we remove it, we end up with this only singlet. So singlet is the cells which the single cell represents the cluster information, all cluster information. So we now have a good cells, and now we need to test. This is a like method in single cell. We need to test this, which is testing the reproducibility, because we were discussing with the ChemTech industry people. They were because we were seeing a lot of variability in the data set they are generating from their own, because they are saying replicate. When we are generating replicate, we couldn't see the similar kind of just cellular information, but we tested that, and we found that they are kindly more reproducible. They are, as you can see, they are all of 6. So 617 and 617, they are aligning all together. So we found that they are highly reproducible. So our analysis end up with a good cell clusters. So we further taken for the downstream analysis. And as you can see here, this is a final 10 cluster we identify in this Surat analysis, and these cell clusters representing the individual cell types. But in the beginning, I do not have the information of this, like what these different colors, cell clusters are representing to. So I took help of a single cell data set from this Drosophila and Aedes, which they have reported some of the marker genes for individual mid-grade cell types, by taking the help from them, like not taking, means I have identified the homologous sequences from them and identify same in the mice Podoptera 617 cell lines, and I could able to annotate them well with the different distinct cell clusters. As we can see, we would able to find enterocyte-like cells, which is EC-like, and the number representing at the bracket is the percentage of cells. Like we could able to find like 4 enterocyte-like cells, and we could able to identify the visceral muscle cells and enteroblast and stem cells, which is 7%, and enteroblast, which is 16%, and enteroendocrine cells like 8%, and visceral muscles and enteroendocrine cells. We use a different marker genes. The marker gene I used for the stem cells was a notch, and enteroblast we have a lot of marker genes, but so I will show you later. And enterocyte we have trypsin as a marker gene. Enteroendocrine cell we have Prospera as a marker genes to label this particular cell types. And after this cluster identification, there is one more step which is called just getting the good principal component. So we ran this using a loop, and I use the principal component value was 20, so that my cluster looks distinct and separated. And this is another way of representing the same information, which is UMAP plot, by using this marker genes. This is some of the marker genes I have used, like as you can see here, EC-like we use 3 oxaloreductase, and this protein turtle is for the enteroendocrine cells, and sodium-dependent neutral amino acid marker genes for representing this enteroendocrine cells, and mushroom body quinone is for representing stem cells, and myosin for representing the visceral muscle cell. So this marker labeling further confirm the cellular identity of the distinct cell clusters. And this is and then once I got this, once I could able to label them as distinct cell clusters, the next step was to see how they are progressing during the differentiation process. So there was a previous biological paradigm that enteroblasts have or differentiate from the stem cells, and enterocyte enterogyne cells have their origins in this enteroblast. So we proved this by using this trajectory analysis, and we use Monocle 3 software, which is a computational framework software which involve adaptive models, which it just help us to associate the gene expression to read help us to relationship between gene expression and pseudotime. So what we do in this one is we artificially giving a pseudotime values to each cell type clusters, and based on the differential expression gene patterns, it automatically create trajectories, and because we know from the literature that stem cell is the beginning of the cells, and from that other cells are differentiating. So we took this stem cell as initial values, and after that we could see that in stem cell we are seeing stem cell progress to enteroblast, and enteroblast is differentiating into other cell type, which is EC like on enterogyne cells. So this analysis is further confirmations of the previous biological paradigm, like the enteroblast differentiate from the stem cells, and the enterocyte enterogyne cells have their origin in the stem cells or the enteroblast. And to further, this is how we confirming every step. So look, once we get the cluster, we are confirming with the markers, then we are confirming with the trajectory analysis. Now we confirming with this KEGG pathway enrichment analysis. These are some of the pathways which got enriched in this what we call different Midgard cell types. And the pathway which got enriched in enteroblast was ribosome, spliceosome, longevity regulating endocytosis, and protein processing in endoplasm. And this signature, this pathways is said to have a play role in cellular differentiation proliferations. Likewise, for the stem cells, the pathway which got enriched was proteosomes, TGF beta signaling pathway, Hedgehog signaling pathways, and WNT signaling pathway, and oxidative phosphorylation. And these are some of the pathways which got enriched in stem cells, and the pathway which got enriched in this enterocyte was ATP-dependent chromatin remodeling, protein export, phagosome, which this has already been reported in the Drosophila. So we further confirm with our analysis with this pathway enrichment. Likewise, as you can see within visceral muscles, and this motor proteins phototransduction pathway set to play a role in muscles and has found to be expressed highly enriched in this visceral muscles in our data set as well. So which further confirm the identity of our single cell data set from the Midgard cell lines. So this is gene ontology pathway enrichment. Further is the gene. Likewise, what I did for the KEGG. The KEGG pathway is a pathway which, as all of you know, what is KEGG pathway, and this is called gene ontology, which involve the biological process, molecular process, and molecular function. These are some of the pathways which got enriched, involve EB, cell divisions, cytoplasmic translations, and stem cells. You were saying most of the pathway is got enriched, and this EC, we have some specific, which is protein catabolic process, oxidative phosphorylation, protein DNA complexity, and these are protein transport transmembrane transport protein pathway, which is said to play an important role in enterocyte, because we all know enterocyte is a secretory cells, and so this is stem cell and visceral muscle cells, and So now, after my confirmations of my single cell data set of my Midgard cell line, that this homogeneous population, which we considering before, is not homogeneous, it is a heterogeneous populations, and further to confirm this identity of this cell types, we would be interested, industry people would be interested to enrich this stem cell specifically for their to testing this insecticidal target. So we are in the process of developing a method for enriching this, and this is I used to confirm my work as well. But in our lab, we have another project which is working on this stem cell differentiation, standardizing the stem cell differentiation and enriching the stem cells projects. But we're currently working on that. But in my work, I enrich the stem cells to standardize the method of enrichment. So I took these cell lines, I grew these cell lines and designed some marker genes, which I showed you before. Notch is a marker gene for stem cells, and we have marker genes like forcat is also the marker gene for stem cells, and prospero is a marker gene for enteroendocrine cells, and esterase is the marker gene for enterocytes. And we designed the what we call the like a construct expressing the promoters of these marker genes, and we transfected these into the midgut cell line using some reagent, which is Myrus, and there is sulfactin also. And so we transfected the cells, and after the transfection of the cells, and we have, in this construct, we have red fluorescent protein and green fluorescent protein. The green fluorescent protein, we used it to test the transfection efficiency, and the red fluorescent protein we used to test whether our insert get inserted into the particular cell types or not. And after like transfection, we grew the cells in the media, and 42 hours transfection, we removed the media, and we've added a fresh medium without any reagents, and we grew them for like 3 days or like 48 hours. And after the 48 hours, we checked them under fluorescent microscopy, and there are two methods I have listed here. First, I go with the FACS, and then second, I go with this the puromycin treatment, because we have in that, I will show you the construct later. And so we followed this method for enrichment on stem cells. The transfected DS red under stem cell specific EGFP, we have this construct DS red is under stem cell specific promoters. And we have this EGFP under HR5, this is element, this is a promoter element, just to so that, as I told you before, yeah. So then once we get, after 48 hours, we saw them under the microscopy. All the cells, some of the cells were showing red color, red fluorescence. These red fluorescence cells, we took them to the FACS facility, and we shot them out using this fluorescent activated cell shorting method. And shorted cells, mostly they are RF positive cells. And once shorted cells, we took out from the facilities and grew them in conditioned medium, which is a medium we usually used for growing this midgut cell lines. On top of that, we have added this 20 ecdiazone this hormone 20 hormones, because this is facilitate literatures have reported they facilitate differentiation in growth. So second method I used for the enrichment of the stem cell was using this puromycin, because in our construct we have puromycin driving the expressions of this DS red, and along with this notch promoters. So we selected this using this different puromycin concentrations. And after that, surviving cells exhibiting RFE fluorescence more than 100% and showing the morphology similar to stem cells were taken further, like I did for this FACS cells, and grew them in the same media in a separate culture set. And this is the construct I use. I talked before. This is notch promoters, having notch promoters, and driving the expressions of DS red monomer to short them. And this is HR5 is the promoter element to driving the expression of EGFP to check the transfection efficiencies. And like I have designed two constructs. First, we would be interested to check whether these construct is working or not. So we designed the first construct, and then we were interested into enriching the cells. So we use this, driving the expression of using the puromycin in selection. And as we can see here, these are the, this is, as I can show you here, this green fluorescent protein showing the transfection, and the red RFC showing the cell types expressing the notch marker genes, promoter of the notch marker genes, and forcat is a promoter for the stem cells, and prospero is a promoter for the marker promoters for this enteroendocrine cells, and esterase is for the enterocytes. So here we can see different, as we proved our hypothesis, what we were identifying in the single cell data set. We proved this, that we have a different heterogeneous populations of the cell types in this midgut cell lines with this work as well. And still currently we are growing these cells, and yes, ma'am. Oh, sorry. Yeah, so I'm going to finish it. So this is, currently we are growing these cells in the medium, and we're still working on the factors that helps us to proliferate and differentiate. We are struggling with this currently. So one of my colleagues was working on that project. So my hypothesis was he's using my construct for enriching the cells, and this is how we grow this monocle cell lines also. We have a plan to grow this monocle cell line stem cells, so we're growing them. They still, after 20 days, it looks better. It doesn't look dead to me, and it is still growing. So, and because I told you before that my work, I'm working with the Camtech, so they were interested to see this insecticidal target, because no information is available currently on insecticidal target in identify in the different mid-grid cell types. So we could able, with the single cell data set, we could able to identify this insecticidal targets and label, identify in the different mid-grid cell types. And as we can see here, distinct cellular differentiation cellular patterns was observed, and aminopeptidase is this. I have done for this respiratory as well midgrid, but here I've shown you the midgrid targets. Aminopeptidase, cadherin, ATP binding cassette, phospholipid transporting ADPase, BBC is a target genes for the midgrid membrane. And we can see their expression in different mid-grid cell types as well, because aminopeptide was shown to be expressed in mostly in enterocytes. Cadherin is in almost in all of the cell types, and there are some, likewise, ATP binding cassette is expressing in different, and phospholipid transporting ABCA was said to be expressed in enterocyte and EE-like cells. So this is a pipeline I have standardized, and now it is currently available in, I have uploaded this pipeline in Camtech website. Anybody would be interested in analyzing a single cell data set can take this pipeline from the Camtech. And this single cell data set analysis pipeline, currently I have included 3 replicates. We can add as many as we could, but by modifying the codes. So these are some of the steps which involved in this analyzing the pipeline. I referred this my application as scRNA-DB for analyzing single cell. These are some of the steps I discussed in the first slides. And this is a SINE application. You don't need to, any non-bioinformatics people can analyze this data set very easily. So I have designed these applications, and you just need to get the filter matrix and just input, just provide the path. Set your, whatever genes you would be interested, like mitochondrial genes, which you want to remove, and the genes which you want to include to see on pictures, like we have TSNE plot, feature plot, and volume plot. We have some of the parameters like N count RNA. These are some of the parameters we can set manually. So you don't need to go into the code and how to change everything. You can do it manually by looking at over this interface. And you can provide, and click on the start analysis. It will show you the time. Here I have shown 3 minutes, but it usually takes more than hours to complete the finished analysis, because I have used the smaller data set to represent it. So anybody would be interested can take this from our labs for analyzing single cell data set. In summary, we have like cell dissociation isolation method were optimized for mid-grid cell lines, and 2 replicates show the good reproducibility, and cell markers for mid-grid cell types were identified, and RA target representing different mid-grid cell types were compared. And a user-friendly scRNA sequence analysis pipeline were also developed, which is SINE and R-enabled, available to every member if you can discuss with us. So relevance is understanding the cellular landscape of the midgrid can be crucial for developing new strategies. This finding exists validate the current biological paradigms and underscore the utility and inherent limitation of immortalized cell lines, further highlighting the invaluable role of scRNA sequence in advancing our understanding and potential improvement of this cellular resources. And special thanks to my supervisor, Dr. Subareddy Palli, and Dr. Doug Harrison, and Dr. Cindy Ma'am for teaching me this cell lines work and giving me this opportunity to present here as well. So thank you so much. Yeah, I'm ready to take questions. Any questions?
We have plenty of we'll see. Can you hear? Okay. Plenty of time for questions. We'll start today.
Thank you. Thank you for the talk.
Yeah.
I stepped out for a little bit, so I missed part of your talk.
Okay.
Which insect species is this that you studied?
This is Spodoptera frugipeda.
Okay.
Yes.
So when you mentioned the RNA-seq protocol,
Right.
you said that the cells were labeled.
Right.
Why? I imagine that I guess you
Cells labeled.
removed the midgut from the insect.
Right.
You dissociated the cells.
We
you-
already have, oh yeah, sorry to interrupt you. Okay, you can go ahead. Those are cell lines.
So why would you have to label the cells before you do scRNA-seq?
I didn't label anything here.
Did you said you labeled
Oh,
us?
yeah, I said I use labeling using marker genes to label the distinct cell clusters. I'm labeling them cell clusters, not the.
Or not the individual cells themselves
Right,
before you
so-
do? I
Yeah,
see.
before you do.
Thank
So because I worked on cell lines first, and currently I finished midgut tissue as well. So yeah.
Maybe one follow-up question. So do you think that insect gut is similar to human gut from your
Yeah.
data sets?
Right.
Very similar?
I have found most of the cell types, what I found in single cell data set from the mammalian cell lines of midgut, and I found most of them. But there was some cells which I couldn't enable because we have pancreatic, different, like, well, not pancreatic, but I mean, there are a lot of different cell types were present in, but I could not able to find out. But in this data set, we could, if this, I have shown you, this is 5 major cell types we found in most of the cell insect cells, and they are representation of mammalian because in the beginning of the single cell from the midgut, they took help from mammalian markers to label them as well.
So they have like goblet cells, endocrine
Goblet
cells,
cells like goblet
planet cells.
Yes, sir.
Okay.
Yeah, but I couldn't find goblet cells in my hair, but I could able to find in my midgut tissue data set.
Okay, thank you so much.
You're welcome.
More questions, Guy?
Thank you for sharing this. It's quite interesting. I started doing work on cells from midguts second half of the 90s, so this more than 25 years ago. It says something about my age. But at that time, there was no single cell sequencing. We have tried together with Marshall Loop and Ray Hakim to identify stem cells by using all the antibodies that were present in the vertebrate field. Nothing worked. So I'm very happy here that you say that you have markers to identify stem cells from the midguts of fall armyworm. So that can be interesting for the field, people working in this field. Now, I have a question for you. The human midgut, there has been a paper published, I think December 22, single cell sequencing of all the cells in the human midgut. If you see the diversity, how many cells are there, how many different cells are there, I think what you present is expected. That there is a high heterogeneity in cells. But what I missed in your presentation is, we all do cell culture, we all do subculturing. What is the impact of subculturing if you use, let's say, a passage of 100 or 250? What would be the difference if you do a single cell sequencing?
What I read in literature, they are saying keep passaging, they are increasing the heterogeneous more.
So
they are more-
the population is becoming more diverse.
Yeah, more diverse, yeah, you can say. I have seen that in like
It's quite
I've read.
interesting.
Yeah, it's very
So you
interesting.
do a kind of, you give some favoritism to some cells that grow better,
Right.
and some other cells they grow less. So that would be quite interesting. And would it then be because some cells are growing faster,
Right.
or they have, let's say, competing properties? That I think would be quite interesting.
Yeah.
Because on your previous question, you say there are nearly no goblet cells.
Yeah.
They're very interesting in caterpillars
Right.
because they control the pH.
Right.
For
But
people working in the vertebrate field, the pH of caterpillars is very alkaline, 9, 10, 11, 12. So this is making caterpillars a unique biochemical incubator, which is completely different from the vertebrate field. A pH of 10, this is crazy. And that would be quite interesting to see if you could sort out and, let's say, favoritize some cell types.
we found this goblet cells in our midgut data set.
Sorry?
We found this goblet cells in midgut from 4, and this what is called the sting bug. We found the goblet.
Okay, yeah.
Yeah, it was.
But yeah, and
I
that
couldn't
brings
find
me,
that in-
sorry, that brings me to another question.
Yeah.
I'm an older person, and when I started this work on cell cultures, it was primary cell cultures.
Yes, sir.
So primary cell cultures, stem cell cultures, you can isolate relatively easy. Takes you a lot of time, a lot of patience, a lot of dissections, but relatively you can isolate quite a lot stem cells. I do not say they are 100% pure,
Right.
because stem cells are always in a kind of a phase from 100% no differentiation to, let's say, 20, 30, 50% of differentiation. But at least if you would do then single cell sequencing of a population that you prepare yourself,
Right.
that would be, I think in your case, a very good addition as, let's say, a population that you have characterized,
Right.
and that would allow you to do some, let's say, further characterization. I think that would be useful.
Right.
And if I may, I have a last one. You talk about replications.
Yes, sir.
And also with my students, we always have a lot of discussion about what is a replication.
It's biological replicates.
Yeah, I know what you mean, biological replication. But what does that mean?
It does mean like
You don't have to answer this now. But you say, yeah, I do my work with 2 biological repetitions.
Yeah.
In fact, as a supervisor, that's meaningless for me.
Actually,
Because what is your certainty? There are here in the room many people in vertebrate toxicology. You can do an experiment with 2 repetitions, 5 repetitions, 10 repetitions. But what do you finally want to have? A result with a certainty. So for that, there is some mathematical programs that say, okay, you do 2 repetitions, you have an outcome with this certainty. You have 3, 4, 5. So there is metrics behind.
Right.
And that would be very useful here.
No,
Single cell
it's like
sequencing is not so
yeah.
expensive anymore.
Okay. Yeah.
Yeah, sorry.
No, I'm saying that we were, in the beginning, we have single replicates of this, what we call 617 cell lines. So we were happy with the 661 replicates. But there were a discussion amongst this ChemTech industry, and they were found that there is a reproducibility, there is a variability in the data set. So we thought if we would, doing the same method for isolation, everything, would it possible that we get the same things all at once? But now I standardize a pipeline. If you have a variability also, we can merge them together. We could take on a single replicate is also fine. So it's not a big deal for us, but it's very challenging technology. It's very costly technology.
My
so we know.
question to you would be, if you would like to have a certainty of, let's say 90%,
Right.
to identify your populations, how many times would you need to prepare an independent sample from your midgut cells of fall armyworm? Because finally, that is how you have to answer the question.
Yes.
And you say 2 or 3, doesn't matter to me how many.
Right.
But did you ever do that calculation?
What I don't understand.
Okay, sorry.
I think you two can talk about this a little bit more
we
later.
Can talk this later. Yeah.
So, great question. Thank you, Guy. Let's give this to Prakash for the final question.
Great work, Sajid. You mentioned that you're doing single-cell sequencing from the midgut tissue
Yes.
of the insect.
Yes.
Just wondering, have you compared the 2 data set from the cell line and the midgut tissue? Have you found differences among those 2 data sets? Because I don't see the data I presented here, so.
So yeah, we my second ChemTech project was on midgut tissue. So I compare data set, single cell data set from midgut tissue to midgut tissue of both the different species. But because I have I have both data set with me, so my current work is on doing the differential things. So I'm doing right now this one. So we have, because cell types are the same. There are one cell type which I was missing in this midgut cell line was goblet cells. Other than that, what I was missing in midgut tissue was visceral muscle cells, and that is not because this is because of my fault, because when we are isolating cells, some cells we are losing out. So it is because of the methods also, but we have the goblet cells in.
Okay. Yeah, why I'm asking is, I think people have worked developing cell lines from midgut for many years, and I think Cindy was great doing that and establishing lines from insect midgut. In literature, it says that the cells lost their identity over passages, and I see your data whenever you're showing the violin plot or the clusters.
Right.
It says EC-like.
Right.
does it mean anything to you, like in terms of identifying EC and EC-like? Is it that it's lost? in
Why I was
ident-
saying EC-like based on the Drosophila paper, and because we have marker genes trypsin, and we do not have, we found this EC-like marker as well as trypsin in the same cluster of the cell, particular cluster of cell types. So I couldn't able to differentiate them, but I could able to differentiate that in my midgut tissue, but not in midgut. That's why because I was not confidently able to say that it is a EC, because I'm finding the marker from EC-like as well. So they are differentiating cells, so we don't know exactly. So I said label them as EC-like to avoid any conflict, any conflict later on. So,
Okay.
but we have this in midgut tissue clearly separate.
Yeah, okay, thanks.
Thank you.
Okay, thank you. Now we have a few more minutes to really open this discussion up to questions for all speakers. So if the speakers would like to at least move forward closer to the podium, and if you have any questions for any of the speakers, this is the time for you to bring those questions forward. So.
So while they're gathering up front, I just had a comment to make based on your comments and what we've seen in like marine invertebrates. And it's just a question for all of you here, because it seems to me like we might be dealing with an issue of stemness. And so, for example, you know, like the mammalian people can get their cells from an organ or a tissue, so you know, or and even with insects, you're from midgut, so you know the source of your tissue. But for those of us in the invertebrate, you know, you're a little bit luckier, I think, Andy, because there are little tissues and organs in the little tunicates. But we've seen this as well, where the cells will de-differentiate and re-differentiate. And so what Surjit had mentioned in terms of seeing these different expressions over time might be, you know, some manifestation of that or something else, and it has implications for what you're doing, what you want, why you're culturing the cells in the first place. And I don't know about Andy, I know for us, we started culturing sponge cells because they produce interesting bioactive compounds, and we wanted to see if we could develop in vitro synthesis. And so you can culture the cells, but you maybe over time they don't produce that compound of interest anymore. So I don't know if anybody wants to address that whole issue of stemness or not, and if you've seen that, like where the cells in culture will de-differentiate and then re-differentiate, because it seems like, Sergey, you were kind of getting, you know, that might be one manifestation of what you're seeing with the single cell sequencing.
Right. You're asking me? She's asking me? Oh, you were asking me? Oh, yeah, we were saying, sorry. My mind was somewhere. Okay,
The question is about, as you continue to grow out the cells, you can at times see them differentiate or de-differentiate,
right.
and you seem to be speaking a little bit towards that. Yeah. Is there anything more you would like to say?
Because I'm not, because I don't have more experience on the growing cell lines. So my experience with the cell lines is to grow them and maintain them. I didn't see, because I was growing them until 60-70%. I just get the population of cells. I grow them all, like looks similar to me. I'm not in vitro biologist and answering this question very well. So my bioinformatics persons, and I just get the cells, see them, I found this thing. So it is the answer for that.
Okay, Michael?
Yeah, I think in our field as well, there's a lot of talk about we like to use muscle cells, and luckily for the cell lines that we have from fish, for the macro cell lines at least, they function as muscle 200 passages in. But I think in the mammalian side of things, especially with bovine cells, which our lab focuses a lot on, we often see a loss of differentiation capacity, even just as much as 5 passages in. I think part of that is oxidative stress, the cells losing their stemness. But I also think, as was mentioned in the previous presentation, there's a diversity of different cell types from an isolation. And I think even if we have 99% muscle cells at the beginning, fibroblasts really like to grow quite fast and overtake that culture, which is something that we've had issues with as well.
I think also this should be addressed in the media formulation. After all, we are here because of media formulations.
Yeah.
So there are some components that you can put in your media, and they will direct your cell type differentiation or trans or it doesn't matter. But also this is connected to the nature of your culture. For example, if you have floating cells like suspensions, and if you have adherent cells, these are two different things. So, for example, for adherent cells, you need, let's say, more proteins and other goodies, let's call it like this, than for floating cells. So it depends on many components that you are adding to your media also.
So one of my lab members was working on this new project, which is called differentiation of the stem cells. They are trying to differentiate this midgut tissue, isolating cell from the midgut and then differentiating them using several factors, which is Loeb et al. Has reported it. So he has tested many factors, but he couldn't succeed in any of those. But what he found in his study was that variation in this FBS helping them to differentiate into different cell types. He's saying when he's adding different concentration of FBS, he's saying different cell types, like I'm saying by morphologically, not like he's not also. So we are doing this currently. So hopefully in the I don't know exactly what to say about it.
I just wanted to bring up also the issue of immortalization that, Michael, you mentioned, because that is of interest to all of us, just really what happens, especially of those that, when it happens spontaneously, what is really going on. And so you presented some data. And one thing I would like to see if anyone else wants to weigh in on that, but I also have a specific question for you, Michael. How did you define when the cells were immortalized? Was it based on passage, or what were you looking at?
Yeah, so in our MAC1 culture, the first macro cell line, we saw what we called a crisis event, where the cells, after I think 40 passages, 100 days of culture, we saw the cells stop growing for about 20 days, pockets of highly senescent cells, and we just subcultured the more proliferative ones out of that. And I think this has been described in literature for mammalian cells as well. So that's what we use to describe our pre and post immortalized. But we haven't seen that for all of our cell lines. So like for the bluefin tuna ones, they've been in culture for a year now, but we've never had a crisis event to really definitively say this is pre and post immortalization. But presumably they're immortal if they've been in culture for so long. So it's a little bit challenging to really define it for every cell type that we have.
Okay, thank you. And any other thoughts just on the whole process of immortalization?
I think I can be heard. It's interesting, immortalization. We talked about this over the break. If you take oh God, now I have to speak softly. Oh yes, sorry. If you take human fibroblasts and you try to make them go into crisis, they don't do that. But rodent fibroblasts, embryonic fibroblasts, go into crisis just the same way. And it's a selection of those cells which are immortalized, in fact transformed, that come out. But that doesn't happen in humans. So it may not be surprising that, for example, the tuna doesn't do this, but the mackerel does. There's precedent in the mammalian cell world as well.
Okay.
I think it's also challenging for us that these fish were wild caught. We don't know how old they were necessarily, and yeah.
They didn't tell you their birth date or anything, no? Okay. Other questions?
Thank you. This is, I guess, more for Sophia and Michael, but anyone else looking into like cellular agriculture. When you're talking about like artificially inducing lipid loading in cells versus differentiating and enhancing like heavy chain myosin, and you're going towards creating like a final product, are you looking at making one cell type that you're like inducing muscle caricature in, but then also lipid loading? Or are you looking at blending two different cell types? Like if you're going from insect cells, you're trying to make them, you know, taste like muscle and fat. Are you doing it differently than you would in like a bovine cell line?
Yeah, good question. I think everything. Since it's so new, I think we're just open to trying a bunch of different protocols to get a mix of cell types. I think in the industry in general, there's a thought of like, you would have your muscle cells, you'd have your fat cells, maybe you'd have your fibroblasts. A lot of people are working with fibroblasts and they taste good. I guess we've tried them, so I can attest that they taste good. So I think, yeah, for what I'm working with, I guess maybe just trying them different and thinking them as different ingredients that we could then, once we understand how to grow them well, we could try different combinations of different things and see what's the sensory profile and that kind of thing. I know Michael's done some work with doing both in one cell type. If you want to talk about that.
Yeah, so for my cell type, we do a lipid loading. It's not an adapogenic differentiation, but it provides an analog to fat that we could use. But again, we're not commercializing products, so I think a variety of approaches are being used: a single cell type, multiple cell types, sheets of cells that you stack together. So I don't think there's one good answer. I do think interesting approaches come when you look at genetic engineering and having inducible promoters for muscle and fat within the same cell type, which I think could be really intriguing.
Cool. Thank you very much.
I have a pretty basic question. This one's for, sorry, go ahead, Andy.
No, it's okay. I wanted to add something to this. So let's call it the first generation in the industry, they started to use different cell types. Now the second generation of trying to make a meat is to combine all these cell types together and to see what's happening. We were the first one who started this at 2006 or something, 2015, 16. And we used to really work on each cell type different and to make media and whatever. And today, in academia field and also in the industry, what happened there, nobody's talking about that. So these are secrets, and this is the technology that they are using. But today, they are trying to find one, let's say, media that is global for all these cell types to work together, and it doesn't work. That's why I will never eat this meat. But this is my thinking about this. Let's say if you have, in one batch, you have a contamination, and you are still doing this meat, okay, to your final process, would you eat it? This is an open question.
Mine was just basic. Anita, you mentioned you had cells in culture without replenishing the medium for a year. How do you a long time? And Sophia, you also mentioned that your predecessor went away for a few weeks and came back. How do you prevent contamination?
Well,
they just didn't get contaminated?
No. They are just happy, you know. I think they're not senescent, but they are kind of resting. I don't know. They are okay. And when we split them again, they kind of get activated and they stretched out and they grow again. They're nice.
Once they're clean, they shouldn't get contaminated.
No.
Well, we've also had insect cell cultures, like especially from nervous tissue, that I've just let it sit in a flask in an incubator for over a year, and all of a sudden it took off. So, I mean, this stuff actually does happen. Okay, the real last question. Here you go.
This is a question about, like, I think I forget the name, but like for the marine invertebrates. So I noticed that the species that you used, you mentioned that it was like one of the earliest forms. It was like it was the first invertebrate of the like before like the vertebrates or something like that. Like, so I'm guessing, like, was that kind of like the first one that kind of diverged from like when I guess invertebrates were kind of like one subset species before they like, I guess, diverged into two separate categories, if that makes sense.
What is the question?
Sorry. So I think, yeah, if you haven't.
I think you're talking about where tunicates diverged into
Yes,
chordate,
yes.
wire tunicates
Right.
and chordate
Right.
through the vertebrates. So I think if you talk about like the embryonic form of a tunicate having a notochord, I think that's going to be where
Uh-huh.
you see the divergence of
Right.
vertebrates and invertebrates. So tunicates being kind of at that
Tunicate.
really important spot.
Yeah.
Sorry, I didn't mean to answer.
Okay. Yeah. I just wanted to ask because, like, I was just wondering, is that the reason why that was the model organism chosen because it could like be used to like potentially investigate for all of the invertebrates in a sense?
Because also when I showed
Right.
the larva,
Right.
it has an autochord, like what you have here, right?
Right. Yeah. Okay, that makes sense. If there's time for. Okay.
It would be nice if that were the reason
Right.
I was chosen, but I don't think a long time ago when that organism was first selected for culture that it was because it was close to vertebrates, right?
No.
Okay.
I just answered the question. Yes, they are, but
Okay.
this is not one of the reasons that it became a key model, you know.
Okay. Interesting. Okay.
It's a nice bridge.
Right. Is it okay if I just have one more quick question? And then I have one quick question about insects. So I'm a plant biologist, so like I was just looking at your presentation, you mentioned that there would be an impact on agriculture. Is it okay if you just go into a little more detail on how like your research would specifically impact the agriculture industry?
Yes, of course. Yeah. Do you want to explain more? Can you
Like
say?
explain more about the impact of your research.
Oh, my research.
Yeah.
Okay. So it's gonna help because currently I'm involved in Chemtech projects, which is industry-based project. They would be interested in the monocle cell lines, specifically to test the insecticidal targets. For that reasons, like we would be interested in enriching the cells, the like stem cells specifically. And likewise, my midgut tissues, they were interested in identifying, because currently we do not have any information of insecticidal target expressing in which cell types. Now we have the information that we can say that, oh, this is midgut targets, which I describe here. It's like aminopeptidase, which was said to be expressed in midgut, but was not. No, we don't know where exactly they are expressing. So this will help to enriching those particular cell types, and we can test them.
Yeah.
We can test them at like a high throughput, like we are doing like currently.
So it's like the specific cell that you have
we can enrich them and we can
okay.
test them. Yeah, that will help. Yeah,
That makes a lot more sense.
Yeah,
Yeah, that's super cool.
Yeah,
Thank you.
so much.
Well, and I think too the overarching answer is a lot of ag chemical companies are very interested in using insect cells as part of the insecticide discovery program. And so, and there's a lot of practical applications that we're going to get into in actually the next session in the afternoon. That's when we'll discuss a lot of the practical applications as to what we've been discussing this morning. So everybody has 1 hour to run, find lunch, and oh, right. So, yeah, we have that media panel discussion that starts at 4, and that's actually going to be in a different room. It's Regency EF, so it's around the corner. So, yeah, please, if you can come here at 12:30, and then the panel discussion begins at 4. Any questions?
Those who may not be here this afternoon, you might want to come back because we will continue this discussion with some of the plant people as well. They're having a session this afternoon that's concurrent with ours, but you might want to come back at 4 o'clock because that'll be joint with the plant cell biologists, and we'll be talking about different approaches to media formulations.
Thank you. Bon appetit, everyone.